All right, everyone. It is my great pleasure to introduce Willow Coyote-Maestas today for today's seminar. Now, Willow first kind of came on my radar, I don't know, maybe 2018, 2019, when I read this just really fantastic, very sophisticated investigation into sort of allosteric hotspots in the potassium channel and how you could query this with sort of library insertion approaches. And I remember thinking, wow, this is like both technically really interesting. It's also like had intellectual ideas about evolution and allostery. And I was like, oh, like who's the postdoc involved? And it was just this grad student, and not trying to belittle in any way. As a graduate student in Dan Schmidt's lab at University of Minnesota. And, it just really had an unusually high level of technical and intellectual combination all coming together in this study. And as I came to learn more about Willow over time, I think a lot of that kind of this blend of rigor and creativity kind of comes from his background and upbringing, where he's sort of a self-described hippie dippy. I found those were your own words in some article I found. Where sort of this bohemian upbringing of, van life and outdoor exploration and being around lots of different kinds of thinkers and minds. And this sort of led him over time, first from an undergraduate at Evergreen State, which is like sort of not an R0 type university, but he found a passion for biology through oceanographic plankton research, as I understood it. Yeah. And then found his way to Dan Schmidt's lab, where it really kind of pioneered a lot of these deep scanning approaches to the study of ion channels. And this eventually led him to, I guess, a pseudo postdoc with Jamie Fraser. I don't really know how to describe it at UCSF that ultimately morphed into a fellow's position and now an assistant professor role. Willow's been honored with lots of different awards and recognitions, including he was an HHMI Gilliam Fellow and then a Hannah Gray Fellow. So really an outstanding scientist. And I think what you'll see from the work he's going to share with you today about how he's kind of taken his ideas about scanning approaches and really applied them to give us kind of this very multidimensional readouts of the pharmacology of different kinds of GPCRs and opioid receptors. So with that, I'll just say, take it away for what should be a really interesting seminar.
Thank you, Scott. That was like the most multifaceted introduction I've had. So you went deep. Or at least you like looked down the Google search and clicked things and read them. But I really appreciate this introduction, and it's been wonderful today to spend time getting to know some of you who I hadn't had the chance to meet, and getting to see people who I care deeply about. And Scott's work was so inspiring to me in graduate school. Like I read he had this paper from his PhD in Wendell Lim's lab about like the emergent potential of allostery proteins. And it was what actually wanted that paper amongst two other papers that my PhD mentor, Daniel, gave to me when I first rotated. And that paper like opened up to me the idea of like, oh, there's this like emergent potential for allostery within proteins. Like, how could we study that? And that was like what I fixated on in my PhD. So then I got to meet Scott, and he's, obviously like clock psychedelic cell man now, but, I was still so excited when he gave a talk at UCSF, and I was totally fangirling, and I'm excited to be here and get to hang out. So truly, and it was exciting that you had read my paper at all when I met you, whatever, last year. I was like, someone actually read that paper, because I thought a lot. People know your paper. But anyways, so I've been at UCSF for a couple of years, for a while now. And my lab, what we're really interested in nowadays is trying to study membrane proteins, broadly defined. And so membrane proteins, of course, underlie kind of the ability for our cells to interact with their physical and chemical environment around them, and that allows us to have complex sensory physiology so we can perceive the world around us. They underlie our ability to think. And then because they're at the interface of the cell in its extracellular environment, the vast majority of drugs either directly activate GPCRs, like 30% of FDA-approved drugs interact with GPCRs, but then also transporters move drugs across membranes for further metabolism. So they're really, really important. And over the last many years, structural biology has really revealed beautiful architectures of membrane proteins. And this for a while, really inspired me. So like Rod McKinnon's work studying potassium channels and revealing the selectivity mechanism, Brian Kabilka's work revealing the architecture of GPCRs has really unraveled how ligands bind to these receptors. And recently, really driven by advances in cryo-EM, we now have structures of many different membrane proteins experimentally. That really started with this temperature sensing ion channel TRPV1, which is kind of my white whale project to understand the temperature sensing mechanism. Which was my intention when I started all this, to solve this problem, which I have not solved. But cryo-EM, as I mentioned, has really exploded over the last many years, and that's revealed the architecture of the vast majority of families of membrane proteins. And so before, it was a brutal time in structural biology of membrane proteins that I wasn't around for. And then, of course, AlphaFold changed the game even more. So now we have models of everything. But really, I would argue that a lot of the fundamental questions in membrane proteins still remain unanswered. As I said, despite being one of the model proteins for structural biologists in ion channel structural biology, cryo-EM, we still don't understand the temperature sensing mechanism of TRPV1. And understanding the basis of efficacy, or how different ligands interact with the same site of a receptor and lead to different functional properties, how that occurs is still really unanswered. And so for me, I've been really fixated on how the sequence of a membrane protein encodes for all the different properties. So the ability for that membrane protein to fold into the right shape, traffic to the right place of the cell, and to explore conformational ensembles dynamically to be able to respond to the environment. I've been very interested in this question. And really from a mechanistic perspective, the ways in which we could answer that question. And so, of course, the way you understand how sequence encodes for these properties, so that you could say, understand the basis of temperature sensation or how ligands interact with the receptors, is by making mutations and then measuring the effects of these mutations on the different parameters you're trying to ask about. But the problem is that membrane protein biology tends to be slow. Studying these properties tends to be slow and laborious. And that's meant that like broad-based models of these properties are limiting. And so the solution we came to a number of years ago was the idea of developing mechanistic mutational scanning in membrane proteins. And that's just like a fancy way of me saying an approach that where we can measure each of these different phenotypes that we're interested in and understand when we make a mutation or when we test a sequence, how is that altering all these different parameters so we can understand how it works and how it breaks and how perhaps it works. And if we can answer this question of like how sequence alter these mechanisms at the biophysical level in the cell and the structural level, we can really understand at a really deep level, how do proteins evolve new functions? How do they break to cause disease? And then how do you develop new pharmacologies that could treat those diseases? And so over the past many years, we've developed a number of methods really focused on creating new ways of making libraries. So with deep mutational scanning, where we make all possible mutations or 10,000, 20,000 mutations in a gene, and then measure their effects with multiple phenotypic assays. So measure the effect on trafficking, measure the effect on activity, and then we can disentangle and understand the basis of, say, biogenesis or specific activity, at least in so far as it's encoded within the sequence of a membrane protein. And so these include, as I mentioned, ways of making libraries that was really helped and pioneered by a postdoc in lab, Chris McDonald. I should have his picture because he's sitting right there. And he is now a staff scientist in Hannah Wayman-Steele's lab, but was awesome to work with, and I deeply miss working with him, but I'm happy you get to work with him and your whole community gets to benefit from him. Anyways, we take these libraries that Chris helped pioneer the building of, and we take those and we put those into stable cell lines. And then we apply multiple different assays to those libraries to pull apart and understand how those work. And we're always trying to develop either fluorescence or survival-based assays. And just to give you an example, because I'm going to show you tons of data soon, I just want to make sure we understand how this works. Pretty much, the idea is we have these libraries in cells, and each cell has one and only one copy. And then we sort out, based on whatever phenotype we have, have the relative effect of that phenotype. So in this case, it's surface expression. Oh, my mouse isn't showing up at all. That's okay. So you can see there's different numbers of membrane proteins over here. I guess I am going to meander over. So like that's unfolded to trafficked, and we can sort that out on a fact sorter. Then we can sequence and effectively then get counts for all the effects in our screens, which then we can go and do statistics on to be able to determine the relative effects of all these mutations. And that allows us to measure the effects of 10,000 mutations at once. Of course, you guys are all here where Watson is, where he's been doing similar approaches to these. So perhaps you're familiar with the concept, but I just wanted to make sure we went through it. And so we've applied these to a number of different proteins, including a GPCR, that was a proton sensing receptor that I'll tell you a little bit about in a second, an ion channel, and a transporter. And we apply these broadly across different membrane proteins so we can try and learn, say, what are the determinants of surface expression, which would be a shared phenotype, or in each specific one, we can understand fundamentals of, or understand how they work and how mutations are breaking them. I'm not going to talk about these stories very much, but because we're almost going to be entirely in GPCR land today, I wanted to, sorry, Gail, but that's not true. But I just wanted to highlight that we were learning similar things across other proteins, and I'm really interested in trying to understand broad-based principles that kind of are universally acting on these proteins. Like, what are the biophysical rules that determine whether a membrane protein gets made or not? And how can we learn those from these experiments? But now we're in GPCR land. GPCRs, of course, are fundamental sensors that allow our cells to recognize and respond to their environment. And so they're a very large family, one of the largest, if not the largest family in us. And they underlie many different types of physiology. And really, there are these incredible examples of evolutions, like diversifying of a single protein, where, as my student Matt says, they're heptahelical bundles of fun, or just 7 transmembrane. Yeah, I totally stole that from him. It's awesome when he said that. But they can bind many different stimuli. So that includes small molecules, which are like easy in our minds to understand how do those work. You just bind there, and well, then some cascade occurs, but also something like protons, like pH. So I'll tell you a story about in a second, where protonation will change some conformational state somehow by altering some subtle movements in a protein. And so that makes them very interesting, because they could perhaps sense force too. And all of these different parameters can be sensed by these proteins. And so, like I said, I'm going to tell you now about a story on a proton sensing receptor. So this receptor is GPR68. And what makes it very interesting is, as I said, like usually a small molecule binds, and that's how we think of a GPCR, and that leads to activation through some conformational change. But with pH, it's an interesting problem because you can't see protons in cryo-EM structure. So it's very hard to know where a protonation event happens. And even if you knew where a protonation event could occur, you don't necessarily know which are functional or not, because many residues are ionizable. So the question, instead of it being like what residues are protonated or not, it's which residues that are protonated are functional. And so we thought this was a unique, and this is Matt, the one who said heptahelical bundles of fun. I'm very lucky to work with him. He's an extraordinary student who like, yeah, I'm very fortunate to work with him. And so we then applied deep mutational scanning with the goal of being able to map in an unbiased way how each residue contributes to proton sensing. And then for assays, we use surface expression and proton activation. And that would allow us to differentiate which mutations affect protein abundance, which is most of the deleterious mutations, and then which affect pH activation specifically. And then what we can do is, there's my mouse. Woo! Is we can then take these scores, and you can see there's almost a linear-ish distribution that's here. And we can then, for each one, so each of these little dots are mutations. And so this allows us to then effectively normalize the data based on proton activation, and then get to an unbiased map of expression normalized activation, or which mutations are affecting activation but not stability. And so that is over here in this heat map, which I'm not going to spend a ton of time on because heat maps are kind of busy to look at. You can just see there's these red columns which tell you these residues are important in pH activation. And so then working with a collaborator, Ashish Manglik, and his student, Nick Hoppy, they had structures that were solved, and we were able to take this data and paint it directly on the structure and develop an unbiased map of pH sensitivity. And you can almost see that there's this cluster of residues. Oh, I have to be looking up here because it's not on here. So there's these cluster of residues that seem to be really enriched in red, which is pH activation is specifically disrupted. You can almost see like a pathway of residues deeper into the receptor, which perhaps is representative of pH events happening somewhere, or protonation events happening somewhere here, and then that leading to conformational change, which will drive activation and release of a signaling protein down here. And so then we look deeper at the receptor, and we can look in, and we see, again, that there's this protonatable residues, so 2 histidines, which of course are protonated at biologically relevant pHs. And then we worked with MD simulations folks to get us predicted inactive state structures because they couldn't solve an experimental structure. And we're able to then even see that there's this, as I said, potential pathway of residues that are leading deeper into the receptor. And this is a canonical switch residue that then goes through a conformational change. So we were able to use mutational scanning really combined with experimental and computational experimental structural biology to be able to reveal proton sensing mechanisms. So this is the end of me talking about published data. This is an exciting story, but we've done this already. So we could do this on lots of proteins, but that's not what I want to do now. And I want to take it a step back. And I want to think about the full complexity of when we make a mutation, what could be happening, and just thinking about a GPCR. So a GPCR can be mutated, and that would alter something like protein abundance in the ER, so biogenesis, and that'll lead to an effective surface expression. And then once it gets to the surface, you have stimuli, and you have this whole process that can occur. You get internalization, recycling back to the surface. And it's really at the surface, perhaps, where you're getting signaling events that lead to specific signaling responses. And we're going to really talk about today going really deep on when a ligand interacts with a receptor, what happens? How does it actually activate the receptor? And how do different ligands interacting with canonical receptors activate it? And so really the question we're trying to ask here that many people have been trying to ask for a very long time, including our collaborator, Ashish, who's solved the first structure of an opioid receptor in his PhD, Mark Vanzastro, who is like an old hat working on opioid receptors for a while. He's an extraordinary cell biologist at UCSF. Really, again, led by Matt. So this is a newer picture of Matt after a couple of years into his PhD, after his first paper. And so what we can see is that when a GPCR activates, of course, you get a dose response curve. And we're really interested in understanding what are the parameters that change, because pharmacologists have really struggled to describe, they'll do a dose response curve, look at EC50 and Emax, and they've struggled to describe the mechanism of action of opioids. So structural biologists have solved now over 20 structures after Ashish's first structures of opioid receptors bound to their ligands. And here I'm showing you many different ligands bound to structures, and all the structures look exactly the same, more or less. And that's because to get a structure, you need to engineer them in a way that you stabilize them in a state, and that always ends up in kind of the same state. And so it's very difficult to understand the differences between, say, a partial agonist and a full agonist, or the ability for the same ligand to bind and lead to less signaling or no signaling, despite binding to the same place. And so, as I said, the field has really struggled to describe how ligands interact, different efficacies interact with the receptor. And so that like implies that we don't have, kind of going back to some of the questions I was really interested in my PhD, we don't have good methods to be able to map kind of this like allosteric property of activation or whatever it is that leads to, say, morphine, which is a full agonist that we all know. You know, it causes huge problems in our society. Naloxone, which is what's in Narcan and is used in nasal sprays, that stops the activation receptors. And if you look at naloxone versus morphine, they're really almost, it's just a subtle difference in an oxygen here and just a little bit longer of a group here. And again, these are somehow binding to the receptor and leading to different activations. And in the middle, you have buprenorphine, which is this like big, hairy, spindly molecule that acts as a partial agonist and is one of our best treatments for opioid use disorders. So we're trying to understand kind of what is it in the receptor that allows these to sense differently. And as I said before, structural biology has really struggled to describe this property thus far. And so the predominant model is one of dynamics, which is that you have multiple different states, and this has been backed up by NMR, single molecule Fred experiments, and EPR. But effectively, you have an inactive and an active state, or a number of states, and effectively, when a ligand binds, you shift the distributions of states towards active states, and then a transducer will bind, and that leads to signaling that selects a conformation from that ensemble, okay? But all of this that has been done thus far is really based on, all based on single molecule Fred or NMR. And that doesn't tell you how residues actually are interacting with receptors. That just tells you the overall states that are present. And so what still remains unanswered is really like, how are the residues interacting with the ligand in a way that's able to sense different parts of the ligand? And do ligands activate distinct intermediate states? Is another question one can ask her. And are there different residue networks or like allosteric networks that are being used or tapped into or harnessed by these ligands? So it's all these sorts of questions that I think get to like weird areas of protein biophysics that I've been thinking about for a very long time. And so what we want to do is use deep mutational scanning or make perturbations in dose response and then map out the effects of perturbations on the actual pharmacological parameters. And if we can do that, maybe we can pull out mutations that tune this in different ways and then be able to pull out the properties. So again, we make a deep mutational scanning library that encodes all single point mutations in it. We put those into cells, and then we apply screens to them that are fluorescent. And that allows us to pull out the effects of mutations overall. And we do this with a signaling assay that I talked about very briefly before, but that's measuring cyclic AMP production through a transcriptional reporter that's fluorescent that we can then, and in this case, the opioid receptor is inhibiting activation, so you're measuring inhibition. And so this is just one example of a heat map mapped onto the structure for surface expression. And in this case, we can look at the structure and we can map the data onto the structure, and we can learn things about like which parts of the protein are important in surface expression. But again, what I wanted to get at is this dose response experiment. This took us years to do, genuinely. Matt is like an incredible trooper to make this happen because we needed to do a lot of boring optimization that I'm going to totally like gloss over. But I just want to say this was like incredibly hard, and I think it's extraordinary that we were able to do this. And so this is just looking at a tiny bit of ligand, a little bit more ligand, more ligand, and more ligand. And so we have now the full mutational profile for how active any given mutation is in the context of all 10,000 mutations. And we have this for 3 different ligands. And so just to give you a sense of what the data looks like, to be like, am I lying? Like, do we have good data or not? So this is looking at a position that isn't important at all in function. And this is just from our sequencing, you can see dosability in a way that looks like the mutations aren't doing anything. So each of these lines that's a different color is a different mutation. This is an example of a residue that binds the ligand, and the synonymous variant that we encode at every single position is shown in blue. So this looks just like that over here. And here we're disrupting activity, but we're actually seeing differences in basal activity, which implies that we're breaking ligand binding, but in some cases, mutations are turning it on a little bit and just giving signaling. This is an example of just some random mutation I pulled out just to show you, like, here's a residue that points at the membrane and many different parameters change, and I have no good explanation for it from this. And we have this for all 10,000 variants, and then we have that for each ligand. And so it took us a really long time then, a couple years ago. So we first got this data, and then we had to go to our friends and collaborators, Harold and Jerome at UCLA, who are statisticians, who helped us develop statistical frameworks so we could get fits out of this. And then from those fits, we can then pull out these different parameters and try and ask the question, what are the residues that are important in this parameter of the receptor? So this is, for instance, looking at E-min or basal activity. I don't know if you remember, but there's an example of the curves that were changing the height and that were totally breaking it. Those are encapsulated by this. When we take those and we map those onto the structure, so now what I'm showing you in blue are residues in which mutations are overwhelmingly tuning basal activity. And what that shows is this one helix, for some reason, is really lighting up. And in fact, there's an inhibitory sodium that binds right behind this, right where this transmembrane is, right behind it. And there's a glutamate that binds to that inhibitory sodium. And if you look at that mutation, it's highly gain of function at that position. And it seems to be some sort of coupled effect across this transmembrane. In fact, there's like a tyrosine that faces in here that we remove it. So our model of what's happening here is we're effectively displacing inhibition, and we're just leading to basal activity. And there's a bunch of different mechanisms. And what was kind of fun is an allosteric modulator came online that was solved, or sorry, not solved, was published recently, that binds right here. And then another one was bound on the other side of the transmembrane too. So we didn't do it at all, but you could imagine using this data to identify sites that could be bound by an allosteric modulator for pharmacology if you care about making drugs. Then we can look at EC50, which is the parameter that controls potency, which is classically thought as a description of binding. We can also pull out Emax. And what I really want to highlight here, for those of you in perhaps Watson's lab who maybe has done similar experiments, and for me that was very surprising, is if I go back and forth between these heat maps look really, really different. And so what that means is we're pulling out different parameters of protein activity. And usually when we do these screens and we measure multiple phenotypes, they're highly related to each other. So it's like one of the first things that made us really excited, that perhaps we're measuring something different by doing this ridiculous experiment. And maybe this ridiculous experiment is actually worth doing, which was a big question for us for a long time. So it's a lot of work. So then this is just showing one drug. So this is the first drug we did this of. And I'm just showing you then EC50, or the concentration it activates at, and then Emax, or the maximal sensitivity of the assay. And on the right here, I'm showing you potency, or the EC50. And what we can see in blue is that it's primarily around the active site up here. And then a little bit down here, which are places that bind G proteins. And then in red, I'm showing you the residues that are important in Emax, or efficacy, that are painted on the structure. And these are distinct residues from the blue ones. And it's not just that they're distinct, they're really, you can see that they're almost like moving down the receptor. And they have like a lot of classic allosteric switches, which is what the GPCR people like to call them. But there's all sorts of residues. And if you kind of look back and forth really quickly with your eyes, which I don't ask you to do, but if you did, you might notice that it's almost like they're moving down the receptor, and the residues that are important in Emax are like almost directly coupled to the residues that are affecting EC50, or it's almost like they're second shell effects, or in some ways that I think is deep and meaningful. But there's a lot of things that are interwoven together here still. But one of the main takeaways that we thought was really interesting is these are really determined or driven by distinct subsets of residues, or there's like this organization within a protein that allows some to be involved in binding and some involved in coupling of some sort. And now we have this of all these different structures, and we're still, if you have really good advice on how to deal with this amount of data, I'm all ears. But like one thing we're looking at, so this is Damgo, which is a peptide agonist, morphine, and fentanyl that we've done this for. And you can see largely they look the same. Most of the residues are similar, but there are very subtle differences that tell you something about the ways in which fentanyl, which activates the receptor more than morphine, how that binds to the receptor. And in all cases, we see EC50 kind of decreases as you go farther from the binding site here. And then you see some, so what I'm showing each of these positions as a residue, and it's the distance that residue is and the overall effect of a mutation with different distances. And you can see that like some residues pop out, and those are down here at the bottom and are involved in activity. You can even see that there's subtle differences between ligands, like here and here, or over here, which implies different ways in which these receptors are engaging the receptor and driving something to do with their downstream signaling partners. We have this for Emacs, which again are primarily in what we could call secondary shell residues or in kind of quote unquote allosteric residues. We can take these, we can start to compare across them and just ask the question, what is the similarity and difference between them? And this is just looking at fentanyl and morphine. And in this case, we see this incredible distribution where they're almost linearly related to each other. This is like one of the most beautiful linear relationships I've ever seen in any of our data, where you get a really nice fit. So the bottom here, these are all dead. These are all very active, like wild type. And then you see these off axis. This implies any mutation over here is specifically affecting fentanyl activation. So disrupting morphine, but not fentanyl, and vice versa. And so we can take this and we can kind of map out a bias, just how does each residue change or is involved in fentanyl versus morphine binding. And then from that, we can look that on the structure. And so anything in blue here is a residue that's more preferentially involved in fentanyl binding. And anything in red is more preferentially affecting morphine binding and activation. And so it's super early days. Like we got this data back just a couple of weeks ago, but I'm very excited to share it with you all because you could imagine we're trying to figure out how to even think about the data and how do you it's kind of like we're like the dog who caught the car, and then it's like how do we find meaning. And then we needed more ligands because we didn't have enough data. But no, seriously, we didn't have enough data, and the reason why is because we just have 3 ligands right now, and to some degree, we wanted to understand, is each ligand in the subtle effects of it, is it its own like special butterfly, or are there consensus effects of different ligands, which implies like we could actually pull out what are the parts that have to do with efficacy or like agonism, like that makes something a partial agonist versus a full agonist or an antagonist. So then we went ahead and did this experiment at saturated amounts of drugs, and at the top are the antagonists, in the bottom are the strongest agonists. And you can see this is the distribution of all the mutations. You can see they almost ordered themselves completely, which was like extraordinary to me. And if you look at these heat maps, you almost see these like stripes of blue at the top, which are mutations that make antagonists into agonists, which is very strange. And at the bottom, those go away. And then you have stripes of red kind of pop up. And what this implies, at least to me, something deep about pharmacology is all these residues are in a continuum. It's not just like these classes we just make up, or like antagonists or partial agonists or agonists. All of them are interacting with the receptor, and there's something like there's something emergent in that interaction that leads to the bias of these different states. But an antagonist is just one mutation away to being an agonist, or one subtle change in the chemical group, and vice versa. And so then we took this data, I'm flattening it based on data set, or based on drug. So these are now the mean effective mutation at each position. And then we just clustered the drugs. And what was shocking, again, is that the drugs clustered based on their impact, their strength, and not based on the chemical structure, which I'll show you in a second. And then we can pull out specific places in the receptor that are important. So in this case, red means deleterious. And so here, TM6, which is known to go through this waggle and activation, really has an enrichment of deleterious mutations specifically for partial agonists, and that have less of an effect in full agonists. And we can go over to transmembrane 1, which is not canonically thought to be involved in activation of GPCRs at all. And we see really profound effects. Some subset are specific to partial or like the weaker ligands and not the strongest ligands, but others are inverted. And there's a much stronger effect in the stronger ligands versus the full. So it's not just that like the data overall is organized such that everything is more and more and more strong effects of mutation that maybe is inverted based on the strength of the ligand. But we actually see opposite effects in different regions, which maybe tells us something. And so again, I just want to highlight that this data really organizes not by chemical scaffold. So these are all the ligands that we measure this on. So you see that there's a bunch of, these are the morphinins, like morphine. And then these are a bunch of other compounds. I mean, the fentanyl is up here. But you see that there's a wide variety of ligands everywhere. And we find that there isn't a clear pattern for what makes one an agonist versus an antagonist. And that's why people have been working on this problem for decades, and why we don't have, like, most of the drugs we have that we use for treating opioid abuse were developed in the 60s just through random drug screens. So I'm just going to show you one where we've looked at very closely because it's fentanyl, which is like very timely, and just highlight that we can pull out different residues that are important, which are highlighted in red here, and really understand these are the G protein binding residues. These are known coupling sites. At the top, we're able to look at the residues in the binding pocket that are important. We're able to pull out secondary shell residues that haven't been previously described as being important, this Y77 that we're very, very interested in. And then there's specific residues on the interface of the receptor, like this K100, that makes no known interaction with any signaling protein that we're really excited about as well. And when we got this data back, it's like one of the first times I felt I was learning biology, or like I was finding things that haven't been described before. And so I was very satisfied. So this is now looking at the top down. This is just looking at the residues in the ligand binding pocket. Again, ordered by strength of ligand. And you see that a bunch of these residues are shared across them. And some of them have no effect, even though they're in the ligand binding pocket. So we can start to pull apart this and have a more mechanistic understanding, perhaps, of how ligands are interacting with each of the receptors. So this like Y77 we find, which we mentioned before, that's a secondary shell residue, seems to have a really strong effect in buprenorphine, which is a partial agonist that's clinically very important. And we see in general that the weaker ligands have much more of an effect in this residue versus others, which I don't totally understand what it means, but it's tuning the activity through changing kind of the behavior of the pocket, but in an indirect way. And the stronger ligands don't need that help. But you find other things like this proline and then this set of residues over here on the side that seems to be more important in the stronger ligands. Again, we see differences in sensitivity that aren't just explained by what a ligand looks like. And so we look deep into the intracellular side, because this is the part of the receptor that binds effectors that drive signaling. And we find residues in which we see loss of function specific to the weak agonists. And this is kind of where we think the story is now going for us, as trying to understand what are the mechanisms that makes a weak agonist, or like a partial agonist. Whoa. I went backwards. I apologize. And so then just overall, just to summarize, we've really found, we looked at the structures, and then we tried to pull apart what residues are involved in canonical states contacts. And so been seen in structures before, versus those that are in latent or non-canonical sites. And we see much, much more sensitivity within these non-canonical sites. And I mentioned this K100 before. We're really interested in it. So then we pulled out the mutations, because and we did classical pharmacology assays, because we weren't 100% sure that we were just like convincing ourselves we learned something new, but like maybe it's just sensitivity or assay or an artifact. So what I'm showing you now are Brett recruitment assays. This is taking one of these signaling proteins and just looking at recruitment of that to the receptor in the presence of ligand or not. And so the dose is the ligand. So I'm showing you fentanyl here, Damgo, which are strong ligands, and then PZM, which is a weak ligand. And then when you look at the top here, we can see that many of these mutations, so this is wild type, which looks like this. And then these mutations subtly shift the EC50, but only the strongest mutations totally break it. So this K100, you do a charge swap, K100 to D, totally break it. Whereas for the K100 to N, which doesn't change, which compared to here, it's not changing the charge, you see effect in EC50, and then you see other mutations are very subtle. And we go down to PCM, and we totally break with both the subtle and the strong mutant, and the same over here for these mutants as well. So in other words, we're able to very selectively alter partial agonist activity versus full agonist activity, which people haven't found mutations that do this before. And so this is really exciting. We find a ton of these. And this K100 is a very interesting residue. So V175 is one of these canonical residues. K100 is a very interesting residue. We're now going to try to do proximity proteomics to try and figure out what is it interacting with, and is that like telling us something about some like some interaction that you can't see in structures. Finally, I just told you a lot of data, so thank you for listening to me ramble about my data. But I want to highlight that all of this involved involved developing a ton of technologies. So this is within our lab. Like I said, there's Chris's face finally. Doing library generation. Justin English and Adam Zam at Utah helped us develop transcriptional readouts. We've developed statistical frameworks to deal with this data. And then we've created this really cool interactive toolkit for Chimera X, because Chimera X is a structure visualization software that's developed on our floor at UCSF. And this guy, Tom, who I think of as Chimera Tom, just like single-handedly has built interactive visualizer, which like our data is getting really big, and to be able to find patterns in it is actually quite difficult, and is our great challenge at the moment. So overall, I've shown you that we've done this dose-response mutational scanning, which is allowing us to really start separating out the molecular basis of efficacy or the activation of the receptor from potency and perhaps the binding of ligands, and really to be able to reveal this property. And we're starting to find specific interactions that determine agonism, or that determine partial agonism versus antagonism versus full agonism. And finally, really a big picture takeaway that we find is that overall, weaker agonists are much less mutationally tolerant, or in other words, it's way easier to break the activation of partial agonists. And there's many more residues that seem to be involved in getting a successful signaling event with a partial agonist than a full. And we're now trying to do a bunch of structural bioinformatics to try and test hypotheses like, is it just a two-state model? Do we see different interactions based on like whether you see contacts and that are present in different states? And I think we're going to have an exciting set of collaborations. But and with that, I'm going to tell you one more story. And I have a second acknowledgement slide. So that's like a project that's almost done. That's like answering a really fundamental question that like I've been working on for longer than any other project focus time, kind of almost, yeah. And we're almost done with it. And so I wanted to give you a project where I have no answers. And I think it's just really weird and really cool and fun and topsy-turvy. And so this is to try and understand the basis of olfactory receptor activation. Olfactory receptors are expressed in your olfactory bulb, and in them, each neuron expresses one and only one olfactory receptor. So they're like extremely specific. And then the model is that each olfactory receptor senses, some sense a couple ligands with different specificity, some sense just one ligand, and it's like thought that they have this olfactory code that the olfactory bulb integrates. Okay, no more real biology. But really a fundamental technical challenge for a really long time to actually testing these sorts of models, in particular which receptors are activated by which olfactory ligand, is that olfactory receptors are notoriously very, very, very hard to express. So hard to express that much of the data, my collaborator Ashish likes to say, is just based on noise. Like much of the field is based on noise, or it's a lot of mouse genetics or fly genetics and insect genetics, where a lot of the work has been done. But there's very little molecular work that's done. Another fun or interesting fact about olfactory receptors is they're under really strong positive selection. So they're fast diversifying. So like some of you may taste cilantro as something really nasty. That's coming from a mutation in an olfactory receptor, which changes your perception of smell. And many of you in the room will have a different set of active olfactory receptors in theory than me, because they're not under any purifying selection. And they're in fact used in population genetics as the outgroup. Like if you talk to a geneticist, they just like want to avoid olfactory receptors completely because there's no ability to see constraint. And then there's these theories from protein evolution space and protein engineering, just kind of like thinking about the ways in which stability guides the evolution of protein. So I want you to keep this in your head as well. And the reason why I want you to keep this in your head is that olfactory receptors, again, are very, very hard to express. And so that implies either there's chaperones within olfactory neurons, or they have a unique membrane environment, or something that helps them get expressed. But I think there's a lot of really interesting evolutionary questions to be asked about a protein family that is incredibly tolerant to expression and isn't under purifying selection. And so our understanding of olfactory receptor at the molecular scale is really limited. And I got really started on this project mostly from an engineering perspective, guided by our great collaborator, Ashish, who's been involved in kind of all the projects I've told you about, because he's a co-mentor of my awesome student, Matt, who you've seen his picture pop up. But because he was trying to solve structures. And so he solved the first structure of an olfactory receptor in a big paper a couple of years ago. And what he did is he looked at what are called ectopic olfactory receptors, or ones in your gut. So not really the ones in your bulb, but those ones, at least this specific one, can be expressed. And so they solved the structure of that. Then, but the rest expressed very poorly. And so then what that group did is they created these consensus sequences of olfactory receptors, where they would take whole families of dozens of olfactory receptors and take and try and identify the consensus of that, which is known to increase stability and expression. And they got structures of those. But the problem is you're making tons of mutations, including mutations in the binding site. And so you can't really answer questions of specificity if your structures, if you're having to engineer them with so many mutations. So again, initially, punchline. Initially, we really tried to study this from the perspective of, can we just find mutants that express well or are active? So we did mutational scans for expression and activity, but starting with no detectable expression. And we find, this is just showing a small subset, a tiny subset of the heat map. So the top is expression. It's a very messy data set. We didn't quite believe it when we first generated it, got it. And the bottom is for a ligand where we got actual decent data, but in almost all cases, it's weighted towards gain of function effects, or in this political environment, gain of activity, gain of expression. And so what we did is we just looked and we tried to pull out mutants that we could express. And we found, indeed, much to our surprise, we could dramatically rescue expression, orders of magnitude in flow cytometry space. And so we went from, here is OR1A1, or cells without the receptor present. This is just the tiny hint of expression. And then we're able to rescue the receptor dramatically with a charge swap mutation. And this is showing we can measure activity. This like tiny hump was where we were starting in our screen. And then we can measure dramatically more activity as well. So we're getting both gain of expression and gain of activity. And so that implies that perhaps this could be used to functionally study olfactory receptors. And just to show you a comparison of a lot of different receptors. So beta adrenergic receptor is like the classical, canonical, very well expressed receptor. Again, this is our starting point. This was their engineered consensus sequence that had dozens of mutations. These are our mutants. These are just like expressed far better, more stably. This is way better than that, and really better than that, right? And so then we asked the question, can we rescue other receptors? Because this happens to be like the only residue in these receptors that are conserved. And so what I'm showing you now is a bunch of these receptors. I guess I have to be over here now. So you can just see a lot of them move up. But in fact, this OR6A2 goes down. That's weird and topsy-turvy. Why would that be? And so overall, we can rescue a bunch of these receptors. Makes no sense because they're like canonically so poorly expressed. We're just making one mutation. And to me, this implies really topsy-turvy epistasis within a family. And something else that I told you before, if a long time ago in the talk, that expression and activity are usually related to each other. Over here, this is the data of expression and activity for two ligands. That's not a line. That's not even a hint of a line. That just looks like noise. But as I showed you, we can pull out real mutants. There's real things in there. So it implies there isn't the same constraint biophysically that expression is limiting the like emergence of activity in our screens. And this is showing two different ligands. One on the y-axis that didn't work very well, but is thought of as a ligand, and one on the x-axis that worked quite well. Again, not a great comparison between them. So then we wanted to ask the question, is it just noise? Are these occurring, are the places, the hotspots that are coming out of this, are they in different places of the receptor? And you see, in fact, that this is the increase of surface expression. And you see transmembrane 1 pops up over here, but doesn't pop up so much over here. Oh, shoot, what am I doing? Here, there's here and here. No, there's red here in places where there isn't over here. And then this transmembrane really pops up in activity and kind of expression. It shows that there's differential constraints in each of these, or there's different patterns that are being kind of sampled. Or in the case of, I'm trying to do this here, we find gain of function effects or gain of activity in the context of one ligand and not another ligand. So there's just like a lot of different patterns that are emerging. And usually we don't see different patterns quite like this. I showed you the opioid receptor data. We did so much work. We ended up with like almost the same residues for most of it with just subtle differences. And in this case, it's very different across each of these. So now what we're doing is we're taking these mutations and redoing the mutational scan to figure out what are all the interactions that are occurring. And we're then trying to rescue large sets of receptors and trying to study and map functional specificity. And then we could apply this in other families to try and understand constraints more broadly. And why is it that we see like inverted effects of a single mutation in a family? And I just think this is an extraordinary model system to understand like expression and activity relationships and the emergence of folding, emergence of function, which I think that we've all been like focused so much on good proteins that express well. And I'm really excited to work on really bad proteins that are hard to study because I have no idea what to predict from these experiments, whereas I now can like kind of predict where our experiments go. So I'm like very excited about this direction. And more broadly, we're just really excited to think about this new pipeline we're developing called Shrimple. Our previous pipeline was called Dimple. Chris came up with this name. But we've been working on ways to map out interaction networks within proteins. We're trying to think about how these interaction networks change across families. And then finally, I'm trying to put this work into the context of more complex disease and understand what are the interactions between genes that are constraining evolution and how does biophysics alter those properties. And with that, thank you. I have had awesome people in my lab and continue to have awesome people in my lab. I have really wonderful collaborators who give me joy to work with all the time. Yeah, happy to take any questions.
Okay. I'll let other people ask questions first.
Thank you so much, Willow. This was so cool. I have a thought experiment that I'm curious if you've done already or thought about with your GPCR and all the drug data and perturbations. If
Probably not, but maybe.
If you pretended that you didn't have your data, but you were going to go do a deep dive with the known structures of GPCRs and minor states and say like Boltz 2, or your favorite quote unquote state of the art pose predictor, would it tell you anything about what particular contacts some of those chemical structures might be making or not? Or I guess like the broader questions are like, do those ligand binding predictive AF based models, are they effective in GPCR space? And like what more does your data say that those don't capture currently?
I actually have done that, so I lied.
Okay.
So the question is, how do like cutting edge models for ligand binding based on AlphaFold like models? So this thing called Boltz 2, or this model, it's like kind of one of the best performing and is relatively easy to run. So like how does machine learning compare? It actually does, to me, a shockingly good job, kind of sort of at sometimes pulling out residues in the pocket, which makes sense. You like see those residues, right? And I would say like state of the art models based on like structural biologists who are look at these proteins like this all the time, pharmacologists, they would come to kind of the same conclusions. There are subtle differences, but it can predict some of the residues we see there in the active site. Outside of that, it does a very bad job. So we did the experiment where we ran Boltz-2 on all the mutations in the screen for like all 15 ligands to see what would happen, because and it doesn't pull out any of the ones outside of that, pretty much. So which again, I think we're learning new biology, but I don't really know what to make of it yet. So
really nice talk. This maybe is more related to the bonus
yeah, the
content
bonus
at
slides.
the end. So the Gottsman lab a number of years ago had this really interesting paper where a silent mutation in a membrane protein, I think it was human P glycoprotein, caused dramatic effects on the protein activity. And the explanation was that translation of the mRNA is coupled with folding in ER processing, as well as the post-translational modifications. I'm wondering if you're thinking about that, and if you've ever done a scan where you've, as controls, you make the mutations, but you also sample all codon possibilities for a given amino acid substitution, because they could have quite different effects on expression.
This is a great question. So the question is how have we thought about the ways in which RNA processing could be responsible for these like super topsy-turvy wonky results, and like how we get so much expression? To me, we haven't done the experiment. I have long tried to convince someone to do that experiment, but instead of just like every single amino acid, do sliding windows of varying scales, so you could like footprint out the residues that might be important in tuning expression. And to me, I think, but I have failed thus far to communicate or to make it cool enough for someone to actually do it.
The Gottsman paper was really interesting, but
yeah,
it was one position.
I'll have to look at it. We've looked at that in our data, but we only include one synonymous variant per position. We see hints of this. So we always see a shoulder, and dependent on the assay, we see a shoulder of gain of whatever phenotype, and it's more sensitive in the abundance or surface expression assays versus the activity ones. In the olfactory receptors, the other ORs, for the olfactory receptors, we see there are some mutations that are very surprising, like a swap in a tail in the N-terminus that ended up, we had a tag on the C-terminus or protein. We removed the tag, that went away, the effects of it. That was really strange to me. That could be explained by an RNA thing. It could be explained by something else. It didn't make any sense because they're so far away from each other. And during biogenesis, you'll end up like how could that be? I think some subset of it will absolutely be something like that. And I think it's something that's been really underexplored in the world of olfactory receptors. Yeah.
Thank you. Judith, I promise I'll give it to you next. Judy. Are the mutations in the olfactory receptors revealing conformations that are natively achieved in the native cells, do you think, or are they just evading quality control machinery to retain them?
I'm going back. I think I was talking to a membrane protein biogenesis person, and they said, I should be really cautious of TM1 effects because we're putting a signal peptide on the N-terminus that's routing this through Sec61, and it could be partially that we're like changing that. And so it's entirely possible that this effect is described by that, especially because we see this really weird behavior where there's like a bunch of aromatics and we're removing aromatics, and somehow that makes it worse, which like makes no sense to me. That could be what's going on here. There's this other whole, which I totally glossed over because I don't totally understand it well enough. I don't think anyone does, but there's this model that like, one of the most interesting questions that we're not really working on is the question of olfactory receptor choice. Like, why is it that you get one and only one receptor in each cell? And it's thought to be this feedback mechanism that's dependent on the activation of the UPR that people did, like UPR transcriptional assays in mouse lines. And then they also think it's based on activation of the receptor itself. And so that implies there's actually a selective pressure to have some unfolded protein response, or to be kind of bad as a protein to involve in olfactory receptor choice. So I think part of the benefit is it might actually be selected to like be really bad at expressing in these cases, because you may not actually need that many olfactory receptors to drive activation. But it's a really good question. Like, is this because we're just putting this in HEC cells and it's not optimized for HEC cells and it's optimized for olfactory receptors? People have thought along those lines of looking for like specific chaperones that are maybe increasing their expression or not. It's unclear. We tried to include it in our screen and it did literally nothing. But I feel like it's a good question that we should be aware of.
I was just curious what the natural function of the opioid receptor is, and if there's a correlation between the pharmacology and its natural ligands in your body.
So the natural function is to be involved in like, I'm gonna get in trouble if I talk to, I'm really not a human whole body scientist. Think about this. Yeah, physiologist, exactly. I can't even think of the word. But it's involved in like feeling pleasure and things like that. Like there are hormones that activate, or peptide agonists that activate them, that are involved in circuits associated with that. It's also involved in different opioid receptors are involved in pain as well, of course. Like it wouldn't, in activating it wouldn't give you so much benefit for pain if you didn't. But to answer your question, do the ligands that are peptide ligands have overlap with the ones that we use? I think like, I'm going to do this thing that's like the worst thing to do in a talk, but I'm doing it. So I'm sorry. I'm not sorry. So it's a peptide agonist version, or like a synthetic one, but that one is a, that's a peptide ligand that's like very similar to the ones in use. So, and then these are all the other ligands, right? And you can almost see that like some of these kind of maybe resemble that. And so people have made ligands that resemble peptide agonists. But something that's interesting I've noticed is that like as a non-chemist, when I look at these look really, really different to me. But then when I look at it on the structure, like the contacts they make aren't that different between them. And a lot of the like scaffolding ends up like, we're just not seeing it in the three-dimensional shape here. And so the contacts they make are actually very overlapping between each of these different ligands.
I guess then are the natural ligands, the natural peptide ligands, do they also cluster by efficacy?
I would expect, in this data, I would expect them to. We haven't run them all. This is the only one we run. The one that we like probably should run someday is endomorphin, which is like the closest thing to the one that's in you and me. That's involved in like apparently pleasure in sitting in the sun. That like people from Northern European places have some variant sometimes that lead them to like enjoy the sun more, but that's maladaptive. There's some like human genetics paper about this. Again, not a physiologist.
Selective for living in snow.
Yeah, exactly.
Well, I think in the interest of time, we got to move on, but let's thank Willow for a fantastic seminar. Really exciting.